First, what are chromosomes made of? Let's think of a chromosome like a well-organized library filled with books of instructions. First, the DNA is like the books in this library. Each book contains thousands of pages of instructions. These are the genes, each coding for a specific trait or protein that our body needs. Now, just like books needs to be stored properly to avoid damage, our DNA is wrapped tightly around histone proteins, which act like spools or shelves. They help organize and compact the DNA so it can fit neatly inside the nucleus of the cell. These histones don't just store DNA, they also control which books can be read, helping regulate gene expression. The centromere is like the spine of a two-volume book. It holds the two arms of the chromosomes together and plays a critical role during cell division, making sure each copy of the library is divided correctly between two new cells. And finally, at the ends of the chromosomes, we have telomeres. Think of them like the plastic tips at the ends of the shoelaces. They prevent the chromosomes ends from fraying or sticking to each other. As cells divide, these telomeres get shorter, which is one of the reasons why aging occurs. So overall, the chromosome is like a compact genetic library, carefully packaged, well organized, and essential for storing and passing on the information that makes us who we are.
Chromosome count and complexity differ among species, with critical implications for inheritance, genetic diversity, and disease resistance. Each species is like a unique puzzle. Some are made of few large pieces, while some are made of lots of tiny pieces. The design and color of each piece would be different, and it concludes the final one life. Now let's compare humans' chromosomes with those of wheat. Humans have 23 pairs of chromosomes, with one set inherited from each parent. They are diploid organisms, meaning they have two copies of each chromosome, one maternal and one paternal. Each chromosome holds information crucial to specific body systems, including eye color, immune function, and metabolic processes. With modern common wheat, Triticum estivum contains 42 chromosomes, resulting from polyploidy, having more than two sets of chromosomes. Ancient wheats like einkorn have only 14 chromosomes, while emmer wheat, hybrid wheat, has 28. Polyploidy in wheat makes it highly adaptable to environmental stressors and enhances its ability to be selectively bred with desirable traits such as yield and baking quality.
A polyploidy is a genetic condition where an organism has more than two sets of chromosomes, unlike most animals, which are diploid. In wheat, we see different types of polyploidy, diploid and tetraploid and hexaploid. Polyploid increases genetic variation, which improves resistance to disease, pests, and environmental stress. However, it can also complicate digestion in humans due to higher gluten contents, especially from the D genome in modern wheat. I attached the real image of polyploidy and also, polyploidy is like having multiple backup hard drive for your data. It is the simple metaphor of polyploidy.
First, why chromosome matter in nutrition and health. Modern wheat is hexaploid, which leads to increased production of gluten proteins. This is great for baking, but harmful for individuals with celiac disease or gluten sensitivity. The extra D genome encodes gliadin proteins, which are known to trigger immune responses. These proteins are linked to digestive issue and neurological symptom. Additionally, an increase in carbohydrate-related genes raise the GI of wheat products. This contributes to obesity and metabolic syndrome. A 2003 Mayo Clinic study found that celiac disease diagnosed in the US have increased fourfold since the 1950s. Scientists attribute this to gliadin proteins encoded in the D genome of modern wheat, which provoke stronger immune reactions. The chromosomal evolution of wheat has direct consequences on human digestive and metabolic health. Chromosomes are like dial in the digestive system. When misadjusted, they can harm our health.
Introduces evolution, adaptation, and human intervention. The evolutionary process of wheat can be compared to that of language. There were many varieties of early wheat, but it started with farmers unintentionally choosing good wheat. Over time, the genetic structure of wheat became stereotyped, similar to how languages were initially diverse, but over time people used promised language systems. The 20th century deliberately combined wild wheat genes to create more efficient wheat. While this wheat was a great help in solving food shortages like green evolution, it also created problems that undermined genetic diversity. This is similar to later linguistic scholars creating standardized language rules, which greatly helped to increase the efficiency of languages, but also reduce the richness of them, such as dialects.